right now. As you listen to these words, millions of cells inside your body are dying. Millions more are being born. Tiny repair systems are fixing damaged DNA, removing broken proteins, fighting invisible threats, working tirelessly to keep you alive. And yet, despite all of this extraordinary activity, your body is slowly changing. Your cells are aging. Your organs are aging. You are aging. The strange part is that scientists are no longer sure aging is something that simply happens. For centuries, we believed growing old was as inevitable as the rising of the sun, an unbreakable law of nature, a fate shared by every living creature. But what if that assumption is wrong? What if aging is not a fixed destiny? What if it is a biological process? And what if biological processes can be changed? Because something remarkable is happening inside laboratories around the world. Scientists have extended lifespan in animals, rejuvenated aging tissues, discovered genetic programs linked to youth and repair, and uncovered clues suggesting that aging itself may be far more flexible than we ever imagined. For the first time in human history, serious researchers are asking a question that once belonged only to myths, legends, and science fiction. Can aging be slowed? Can it be reversed? And could the first person to live far beyond what we currently consider possible already be alive today? Tonight, we will explore one of the greatest mysteries ever faced by humanity, a mystery hidden inside every cell of your body, a mystery that connects biology, medicine, philosophy, and the future of our species. Because the question is no longer simply why we grow old. The question is, what happens if we learn how not to? Imagine waking up tomorrow and discovering that aging has stopped. No more wrinkles, no weaker muscles, no declining memory, no biological clock counting down toward death. Would you choose immortality? Or would life lose something essential without an ending? Today, science is confronting a question that philosophers, kings, alchemists, and religions have wrestled with for thousands of years. Why do we age? And more importantly, can aging itself be slowed, stopped, or even reversed? Fun 1, the greatest mystery of life, why must we grow old? Imagine waking up tomorrow morning and discovering that aging has stopped. Not slowed down, not delayed, stopped. You walk to the mirror. The wrinkles that appeared last year have not deepened. Your muscles feel just as strong as they did yesterday. Your memory is sharp. Your energy is unchanged. The invisible clock that has been ticking since the day you were born has suddenly gone silent. No countdown, no decline, no biological expiration date. Would you celebrate? Or would you feel something unexpected? If there were no ending, would life still feel precious? For thousands of years, human beings have asked the same question. Why do we grow old? Why does every living creature seem to move along the same path? Birth, growth, maturity, decline, death. Why does nature build magnificent bodies only to slowly dismantle them? Why does the brain that learns, dreams, and creates eventually begin to forget? Why does the heart that beats billions of times throughout a lifetime eventually weaken? And perhaps the most important question of all, does aging have to happen? Tonight, we are going to explore one of the greatest mysteries in all of science, not just why we age, but whether aging itself might one day be slowed, treated, or even reversed. By the end of this journey, you will understand what modern biology has discovered about the hidden mechanisms driving aging inside every cell of your body. You will discover why some people seem to age rapidly while others remain remarkably youthful. You will learn about the newest scientific theories that challenge everything we once believed about growing old. And perhaps most importantly, you may begin to see aging in a completely different way. Before we continue, I want to ask for a small favor. If you enjoy deep explorations of science, health, longevity, and the biggest questions about human existence, please consider liking this video and subscribing to the channel. This channel is still in its early stages, and every like, every subscription, and every moment you spend listening genuinely helps more than you might imagine. Thank you for being here. Now let's begin our journey. The story of aging is far older than science. Long before microscopes, long before laboratories, long before anyone knew what DNA was, human beings were already obsessed with the possibility of eternal life. Ancient myths from nearly every civilization contain stories about immortality. In ancient Mesopotamia, one of humanity's oldest recorded stories tells of a king searching desperately for everlasting life. In ancient Greece, legends spoke of gods who never aged and heroes who sought divine powers. Across Asia, Europe, Africa, and the Americas, stories emerged about magical waters, sacred fruits, hidden lands, and supernatural forces capable of defeating death. The dream appeared everywhere, separated by oceans, separated by languages, separated by thousands of miles. Yet somehow humanity kept imagining the same thing: a life without aging, a life without death. Why? Because aging is deeply personal. Every human being eventually encounters it. At first, aging feels invisible. Children rarely think about it. Teenagers often feel immortal. Young adults imagine the future stretching endlessly ahead. But slowly, subtle changes begin to appear. Perhaps a few gray hairs. Perhaps longer recovery exercise. Perhaps a photograph that somehow looks older than expected. Then the years continue passing. Parents age, grandparents age, friends age. Eventually, we realize something unsettling. Time is not merely passing around us. Time is passing through us. And no matter how successful, wealthy, intelligent, or powerful someone becomes, nobody escapes entirely. Even the most powerful rulers in history feared aging. Some emperors dedicated enormous resources to finding immortality. One of the most famous examples comes from ancient China. The first emperor of a unified China, often remembered for his immense ambition and power, became obsessed with the possibility of eternal life. He sent expeditions across distant seas searching for mythical lands where immortals supposedly lived. Alchemists promised magical substances that could defeat death. Court advisors claimed they possessed secret knowledge capable of extending life indefinitely. The emperor pursued these promises relentlessly. Yet, despite commanding vast armies and ruling millions of people, he could not command time itself. History repeated this pattern again and again. Kings searched for magical elixirs. Alchemists experimented with mysterious formulas. Explorers crossed oceans chasing legends. One of the most famous legends was the Fountain of Youth. According to the myth, somewhere in the world existed a miraculous spring whose waters could restore youth and vitality. The details varied depending on the culture telling the story, but the underlying dream remained the same. Drink the water, reverse aging, begin again. Today, the idea may sound naive, but perhaps we should not dismiss these stories too quickly, because hidden inside them was a remarkable intuition. Human beings sensed that aging might not simply be a spiritual mystery. They suspected it might have causes. And if aging has causes, perhaps it can be influenced. Perhaps it can be changed. For most of history, however, nobody understood how. Aging was viewed as fate, an unavoidable law of existence, like gravity, like sunrise. Like the changing seasons. You were born, you grew older, you died. That was simply how the universe worked. Then science began changing the conversation. The transformation did not happen overnight. It emerged gradually through centuries of observation and discovery. Scientists began studying cells, then genes, then DNA, then proteins, then the astonishing machinery operating inside living organisms. And what they found was extraordinary. The human body is not a static object. It is a dynamic system undergoing constant maintenance. At this very moment, countless biological processes are occurring inside you. Cells are communicating, proteins are being assembled, DNA is being copied, damaged structures are being repaired, energy is being generated, waste is being removed. Life is not passive, life is active, relentlessly active, every second of every day. The more scientists learned, the more surprising aging became, because aging did not appear to be caused by a single event. There was no single aging organ, no aging molecule, no aging switch hidden somewhere inside the body. Instead, aging appeared to emerge from countless interconnected processes occurring simultaneously. Tiny changes accumulating over years, then decades, then a lifetime. Imagine a magnificent city. Every road, bridge, power station, water pipe, and building requires maintenance. If maintenance is perfect, the city remains functional. But if repairs become slower than damage, problems begin accumulating. A crack forms in one bridge, a power station becomes less efficient. A water pipe begins leaking. Individually, each problem seems small, but over time, the entire city gradually changes. Something similar appears to happen inside our bodies. Aging is not a single catastrophe. It is the accumulation of countless small failures, failures that build slowly, almost invisibly, until one day they become impossible to ignore. This realization has transformed modern aging research. Scientists increasingly view aging not as a mysterious curse or predetermined destiny. Instead, many researchers now see aging as a biological process, and that distinction changes everything. Because biological processes can be studied, they can be measured, they can sometimes be modified. Consider diseases that once seemed inevitable. Infections once killed millions because people did not understand bacteria. Heart disease seemed mysterious until researchers uncovered its underlying mechanisms. Countless medical breakthroughs began with a simple shift in perspective, from accepting a problem to understanding a problem. Many longevity researchers believe aging may be following the same path. This does not mean immortality is around the corner. It does not mean death will disappear. And it certainly does not mean scientists have solved aging. Far from it. In many ways, we are only beginning to understand the complexity involved. Yet the questions being asked today would have sounded impossible only a few generations ago. Can aging be slowed? Can healthy lifespan be extended? Can damaged tissues be rejuvenated? Can biological age move backward? Can some aspects of aging actually be reversed? These questions are no longer confined to myths and legends. They are being investigated in laboratories around the world, and the answers are becoming increasingly fascinating. But before we can understand how aging might be influenced, we must first understand what aging actually is. What is happening beneath your skin right now? What invisible processes are keeping track of time inside your body? Why can two people born on the same day look dramatically different decades later? Why does one person remain energetic into old age while another experiences rapid decline? The answers begin at a level so small that it cannot be seen with the naked eye. Deep inside every organ, inside every tissue, inside nearly every cell, there are microscopic systems quietly recording the passage of time, tiny biological clocks, tiny accumulations of damage, tiny changes that eventually shape the course of an entire human life. And this leads us directly to one of the most surprising discoveries in modern science. Your chronological age may not be your real age at all, because while the calendar measures how many years you have lived, your body may be telling a very different story. And in the next part of our journey, we will explore the remarkable difference between chronological age and biological age, and why two people born on the exact same day can end up aging in dramatically different ways. Before we continue, take a moment to think about your age. Not how old you feel, not how old people think you are, not the number printed on your driver's license, simply your age. Perhaps you are 25, perhaps 40, perhaps 60, perhaps older. Most of us assume that number tells us something important about our health. After all, birthdays seem like a natural way to measure the passage of life. Every year, we add another number. Every decade, we move into a new chapter. The calendar keeps perfect track of how long we have existed. But what if the calendar is telling only part of the story? What if two people who are both 50 years old are not truly the same age at all? What if one person's body is functioning like that of someone 35 years old, while another person's body behaves more like someone 70? This idea may sound strange at first, yet it is one of the most important discoveries emerging from modern longevity science. The age written on the calendar and the age written inside your biology are not necessarily the same thing, and understanding this difference may completely change how we think about aging. Scientists use two different terms. The first is chronological age. The second is biological age. Chronological age is simple. It is the number of years that have passed since your birth. If you were born 40 years ago, your chronological age is 40. No matter how healthy you are, no matter how sick you are, no matter how fast or slowly your body is aging, chronological age moves forward at exactly the same speed for everyone. 1 year is 1 year, no exceptions. But biological age is something entirely different. Biological age attempts to answer a more interesting question. How old is your body actually behaving? Not how many years have passed, but how much wear and tear has accumulated, how effectively your cells are functioning, how resilient your organs remain, how much biological damage has built up over time. Imagine two cars manufactured in the same year. One spends decades parked safely in a garage. It receives regular maintenance. The engine is carefully serviced. The body is protected from harsh weather. The owner drives responsibly. The second car experiences a very different life. It travels rough roads every day. Maintenance is neglected. The engine is stressed constantly. The body is exposed to extreme conditions. After 20 years, both vehicles are technically the same age. But would anyone claim they are in the same condition? Of course not. One may perform almost like new. The other may be nearing failure. Something similar happens inside the human body. Chronological age measures time. Biological age measures condition. And condition can vary dramatically between individuals. This realization helps explain something many of us have noticed throughout life. Think about people you know. Have you ever met someone in their 70s who seems full of energy? Someone who walks quickly, learns new skills, travels, exercises, laughs frequently, appears decades younger than their age. At the same time, you may have encountered people much younger who seem exhausted, fragile, and physically older than expected. The difference is not merely appearance. Scientists increasingly believe that these observations reflect genuine differences in biological aging. Some bodies accumulate damage more slowly, others accumulate damage more rapidly. The fascinating question is why? Why do people age differently? The answer begins with genetics. Each of us inherits a unique biological blueprint. Certain genetic variations may influence how efficiently cells repair damage. Some people inherit genes associated with stronger cardiovascular health. Others inherit advantages related to metabolism or immune function. Researchers have identified numerous genetic factors connected to longevity. Yet genetics tells only part of the story. In fact, scientists estimate that genes explain much less of lifespan variation than many people assume. A person may inherit favorable genetics and still experience poor health outcomes. Likewise, someone without exceptional genetic advantages may enjoy remarkable longevity. This is where lifestyle enters the picture. Every day, countless choices influence biological aging. The foods we eat, the amount we move, the quality of our sleep, our exposure to sunlight, our habits, our routines, these factors interact with our biology continuously. Consider physical activity. The human body evolved in an environment where movement was unavoidable. Walking, climbing, carrying, hunting, gathering. Movement was woven into daily survival. Today, many people spend most of their waking hours sitting. Hours become days. Days become years. The body gradually adapts to inactivity. Muscles weaken. Metabolic health declines. Cardiovascular fitness deteriorates. The effects accumulate silently. Nutrition exerts similar influence. Cells require raw materials to function properly. Proteins, healthy fats, vitamins, minerals, countless molecular building blocks. When nutritional needs are consistently met, biological systems often perform more effectively. When diets become dominated by highly processed foods and excessive calories, the consequences may appear years later. Then there is the environment. Many people think of aging as something happening solely inside the body, but our surroundings constantly shape our biology. Air quality, water quality, noise levels, exposure to toxins, access to green spaces, even socioeconomic conditions, all contribute to the aging process. A person living in a polluted environment may experience different biological pressures than someone living in cleaner conditions. Over decades, these differences can become significant. Yet one of the most powerful influences may be something less visible: stress. Modern life often treats stress as normal. Deadlines, financial worries, relationship conflicts, constant notifications, information overload. Many people live in a state of near-continuous psychological pressure. The body responds to stress through complex biological systems that evolved to help us survive immediate threats. In short bursts, stress responses can be useful. They increase alertness, mobilize energy, prepare us for action. The problem arises when stress becomes chronic, when the body remains in a prolonged state of physiological vigilance, month after month, year after year. Scientists increasingly recognize that chronic stress may accelerate aspects of biological aging. The burden is not merely emotional, it becomes biological. And closely connected to stress is another factor often overlooked: sleep. Sleep is not simply rest, it is active maintenance. While we sleep, countless repair processes occur throughout the body. The brain clears waste products, hormonal systems reset, tissues recover, immune function is regulated, memory is consolidated. Night after night, sleep helps preserve biological resilience. When sleep becomes chronically disrupted, these maintenance processes suffer. The effects may not appear immediately, but over years, inadequate sleep can contribute to accelerated aging. Taken together, genetics, lifestyle, environment, stress, and sleep create a complex picture. No single factor determines biological age. Instead, aging emerges from the interaction of many influences acting simultaneously. This brings us to one of the most exciting developments in modern science. For centuries, biological age was largely invisible. Scientists suspected it existed, but measuring it accurately seemed impossible. Today, that is beginning to change. Researchers are developing tools capable of estimating biological age with increasing precision. One of the most fascinating involves something called epigenetic clocks. To understand this concept, imagine your DNA as an enormous library. The genetic information remains relatively stable, yet not every book is being read at the same time. Cells constantly regulate which genes are active and which remain quiet. These patterns of regulation change throughout life. Scientists discovered that certain chemical markers attached to DNA shift in highly predictable ways as people age. By analyzing these markers, researchers can estimate biological age. Sometimes the results align closely with chronological age. Sometimes they do not. Some individuals appear biologically younger than expected. Others appear biologically older. These measurements have become some of the most powerful tools in aging research. They offer a glimpse into the hidden passage of biological time. Epigenetic clocks are not the only approach. Scientists also examine blood biomarkers. Blood contains an extraordinary amount of information about the body's condition: inflammation levels, metabolic health, immune function, hormonal balance, markers of cellular stress. By analyzing combinations of these signals, researchers can build increasingly sophisticated estimates of biological age. New technologies continue emerging: advanced imaging, protein analysis, metabolomics, artificial intelligence systems capable of detecting subtle aging patterns invisible to human observers. The field is evolving rapidly. What once seemed impossible is becoming measurable, and measurement changes everything. Because once something can be measured, it can potentially be improved. To understand just how dramatic biological aging differences can become, imagine a pair of identical twins. At birth, their DNA is nearly indistinguishable. Their genetic blueprint is essentially the same. In many ways, they begin life with remarkably similar biological potential. Now imagine their lives slowly diverging. The first twin exercises regularly, maintains strong social relationships, sleeps consistently, manages stress effectively, consumes a balanced diet, avoids smoking, stays physically active throughout adulthood. The second twin follows a different path: chronic sleep deprivation, poor nutrition, minimal exercise, high stress, smoking, long-term inactivity. Years pass, then decades. Eventually, both twins reach middle age. Chronologically, they are identical, the same birthday, the same number of years alive. Yet biologically, they may appear surprisingly different. One looks vibrant, the other appears far older. One retains strength and energy, the other struggles with declining health. Their calendars match. Their biology does not. This scenario is not merely theoretical. Studies involving twins have repeatedly demonstrated how dramatically life circumstances can influence aging trajectories. Genes matter, but experience matters too. Every day, the body records information. Every meal, every night's sleep, every stressful event, every workout, every exposure to pollution, every healthy habit, every unhealthy habit. The record is written not in a diary, but in biology itself. And perhaps that is the most empowering insight in all of longevity science. Aging is not completely fixed. We cannot stop time. We cannot choose our genetics. We cannot avoid every source of damage. But many aspects of biological aging appear responsive to how we live. The story is still being written, every day, every year, every choice. Yet this raises a deeper mystery. If biological age can drift away from chronological age, if some people age faster while others age more slowly, if the body is somehow keeping track of accumulated wear and tear, then what exactly inside us is keeping score? What biological mechanism is counting down the lifespan of every cell? To answer that question, we must travel even deeper into the microscopic world. Because hidden within nearly every cell of your body are tiny structures that act like biological sand timers, and their gradual shortening may reveal one of the most important secrets of aging. In the next chapter, we will explore telomeres, the protective caps attached to your DNA that may help determine how long your cells can continue dividing before time finally catches up with them. Part 3. Telomeres, the sand timers attached to every cell. Imagine that every cell in your body carries a tiny clock. Not a clock with hands, not a clock hanging on a wall, but a microscopic biological timer that quietly measures the passage of life. A timer that begins counting down before you are even born. A timer so small that it exists deep inside your DNA. And a timer that may help explain why aging happens at all. For decades, scientists searched for mechanisms capable of tracking biological time. How does a cell know whether it is young or old? How does it know when to stop dividing? How does the body prevent cells from reproducing endlessly? The answer led researchers into one of the most fascinating regions of modern biology, a place hidden inside nearly every cell of your body, a place where tiny structures known as telomeres silently record the history of your life. To understand telomeres, we must first understand chromosomes. Every human body is built from trillions of cells, and nearly every one of those cells contains a complete copy of your genetic blueprint. This blueprint is stored in DNA. If you could stretch out all the DNA inside a single cell, it would be astonishingly long. Yet somehow that enormous amount of genetic information must fit into a microscopic space. Nature solves this problem through elegant packaging. DNA is tightly coiled and organized into structures called chromosomes. You can think of chromosomes as storage containers for genetic information. Inside them are the instructions for building and maintaining your body. The color of your eyes, the structure of your muscles, the functioning of your immune system, the countless biological processes that keep you alive. Every chromosome contains valuable information so important that protecting it becomes a matter of survival. And this is where telomeres enter the story. At the ends of every chromosome are special protective regions called telomeres. These structures do not contain instructions for building organs or producing proteins. Instead, their primary role is protection. They act like biological shields, guardians standing at the edges of your genetic material. To understand why this protection matters, imagine an ordinary shoelace. At the end of every shoelace is a small plastic tip. Most people rarely notice it, yet without that plastic tip, the lace would gradually unravel, the fibers would fray, the structure would weaken. Eventually, the shoelace would become difficult to use. The plastic tip prevents this from happening. It protects the integrity of the lace. Telomeres perform a remarkably similar function. Scientists often describe them as the plastic tips of chromosomes. Just as shoelace tips protect the ends of a lace, telomeres protect the ends of DNA. Without them, important genetic information could become damaged during cell division, and cell division is happening constantly. Every second, millions of cells throughout your body are dividing. Skin cells, blood cells, cells lining your digestive system. Old cells die, new cells replace them. This continuous renewal is one of the reasons life can persist for decades. But cell division comes with a hidden problem. Each time a cell copies its DNA before dividing, the copying process is not perfect. The cellular machinery responsible for replication cannot completely duplicate the very ends of chromosomes. As a result, a tiny portion of the telomere is lost during each division. The loss is incredibly small, far too small to notice. Yet it happens again and again and again. Every division removes a little more. Imagine a candle burning slowly. The flame may appear unchanged from one moment to the next, but over time the candle becomes shorter. Or imagine an hourglass. Individual grains of sand seem insignificant, yet eventually the upper chamber empties. Telomeres behave in a similar way. With each cell division, a small amount disappears, gradually, relentlessly, year after year. Most of the time, this is not a problem. Young cells possess relatively long telomeres. There is plenty of protective material available, but eventually, after enough divisions, telomeres become critically short. And when that happens, the cell receives a powerful message. Stop dividing. This discovery led scientists to one of the most important concepts in aging research, the Hayflick limit. For much of scientific history, researchers assumed cells could divide indefinitely under the right conditions. That assumption turned out to be wrong. In the early 1960s, a scientist named Leonard Hayflick made a remarkable observation. Normal human cells could divide only a limited number of times. After a certain point, they stopped. Not because nutrients were lacking, not because the environment was hostile. They simply reached a biological limit. The cells entered a state where division ceased. This finding challenged long-held assumptions about life itself. Cells were not immortal. They possessed a built-in lifespan, a finite reproductive capacity. Today, scientists understand that telomere shortening is one of the major reasons this limit exists. As telomeres shrink, cells move closer to the point where further division becomes impossible. In a sense, telomeres function like biological sand timers. Each division allows more sand to fall. Each year brings the timer closer to empty, and eventually, the cell reaches the end of its replicative life. This mechanism serves an important purpose. Without limits, damaged cells might continue dividing uncontrollably. Cancer risk could increase dramatically. The body needs safeguards. Telomere shortening is one such safeguard. It helps prevent unlimited cellular reproduction, yet this protection comes with a cost. As more cells reach their limits, tissues become less capable of repair and renewal. The body gradually loses resilience. Recovery becomes slower. Regeneration becomes less efficient. Signs of aging begin accumulating. For many years, scientists believed telomere shortening was simply an unavoidable consequence of life. Then a groundbreaking discovery changed everything. The story begins with a scientist named Elizabeth Blackburn. During her research, Blackburn became fascinated by the structure of chromosome ends. Together with fellow researchers, she investigated how cells protected their DNA. What she discovered would eventually transform aging biology. The team identified an enzyme called telomerase. Telomerase performs a remarkable function. It can rebuild telomeres. It can add genetic material back onto chromosome ends. In other words, it can partially restore what cell division removes. The implications were enormous. If telomeres act like biological clocks, telomerase appeared capable of turning those clocks backward, at least to some degree. This discovery fundamentally altered how scientists viewed cellular aging. For the first time, the biological timer did not appear entirely fixed. There seemed to be mechanisms capable of maintenance and repair. The significance of this work was so profound that Elizabeth Blackburn later shared the Nobel Prize for her contributions to understanding telomeres and telomerase. The discovery sparked enormous excitement. Some researchers wondered whether activating telomerase might extend lifespan. Could aging be slowed? Could tissues be rejuvenated? Could telomeres be restored indefinitely? The answers proved more complicated than many initially hoped. While telomerase can maintain telomeres, excessive activation may increase cancer risk because cancer cells often exploit telomerase to achieve near-limitless growth. Nature, once again, revealed itself to be a delicate balancing act, protecting against aging, protecting against cancer, maintaining life while preventing uncontrolled growth. There are no simple solutions, only trade-offs. Yet telomere research opened an entirely new field of investigation, and it raised another fascinating question. If telomeres shorten over time, can lifestyle influence how quickly that shortening occurs? Scientists have spent years exploring this possibility. The evidence suggests the answer may be yes. Not completely, not dramatically enough to stop aging, but enough to matter. Research has linked chronic stress with accelerated telomere shortening. Individuals experiencing prolonged psychological stress often show signs of increased biological aging. The body appears to translate emotional burdens into biological consequences. Sleep may also play a role. Consistently poor sleep has been associated with less favorable aging markers in numerous studies. Physical activity appears beneficial as well. People who remain active throughout life often demonstrate healthier biological aging profiles. Nutrition may contribute too. Diets rich in whole foods, vegetables, fruits, and healthy fats are frequently associated with better long-term cellular health. Meanwhile, smoking has repeatedly been linked to accelerated telomere shortening. The message emerging from the research is both encouraging and humbling. We cannot fully control aging. We cannot freeze time. We cannot eliminate every source of cellular damage. But our daily choices appear capable of influencing how rapidly biological wear accumulates. The body is not merely a passive victim of time. It responds continuously to the conditions we create. Every night of quality sleep, every nutritious meal, every walk, every workout, every effort to reduce chronic stress. These choices may not make us immortal, but they help support the systems that keep our cells functioning effectively. Still, telomeres are only part of the story. They are important, but they are not the sole cause of aging. In fact, some scientists caution against viewing telomeres as a master key that explains everything. Aging is far more complex. Many biological clocks are ticking simultaneously. Many forms of damage accumulate throughout life. Many interconnected systems influence how we grow older. Telomeres are one chapter, a fascinating chapter, but not the entire book. And as researchers looked beyond telomeres, they discovered another surprising problem. A problem that becomes increasingly common with age. Sometimes cells reach the end of their useful lives. They stop dividing. They stop functioning properly. Yet instead of disappearing, they remain, lingering inside tissues, occupying space, creating trouble, like abandoned factories leaking pollution into an otherwise healthy city. These aging cells refuse to leave, and their presence may accelerate the aging process throughout the body. Because telomeres are only one clock, another threat emerges when old cells refuse to die. Part 4, the rise of zombie cells. Imagine walking through a beautiful city. The streets are clean, the power grid is functioning, the factories are productive, the transportation systems operate smoothly. Everything works together in remarkable harmony. Now imagine that one factory shuts down. It no longer produces anything useful. Its machinery has stopped, its workers have left, the building serves no purpose. Under normal circumstances, the city would demolish the abandoned factory and replace it with something new. But imagine that the factory remains standing, year after year, decade after decade. Not only does it remain, it begins leaking toxic chemicals into the surrounding neighborhoods. Nearby buildings become damaged, roads begin deteriorating, air quality worsens, other factories start malfunctioning. The abandoned structure is no longer merely useless. It has become actively harmful. Something remarkably similar may be happening inside your body right now. And according to many scientists, it may be one of the major drivers of aging. These biological troublemakers have earned a memorable nickname: zombie cells. The scientific term is senescent cells. But zombie cells captures the idea perfectly. They are cells that are not truly alive in the way healthy cells are, yet they are not dead either. They exist in a strange state somewhere in between, unable to continue their original function, unable to divide, unable to contribute normally, yet refusing to disappear. For decades, researchers largely overlooked these cells. They were considered little more than biological curiosities. Today, they are at the center of one of the most exciting areas in aging science. Because many researchers now believe that the accumulation of zombie cells may help explain why aging accelerates over time. To understand why, we must first answer an important question. What exactly is a senescent cell? Earlier, we explored telomeres and the Hayflick limit. We learned that normal cells cannot divide forever. Eventually, after enough divisions, they reach a point where further replication becomes impossible. This is one pathway that can trigger cellular senescence. But it is not the only one. Cells can enter senescence for many reasons: DNA damage, oxidative stress, radiation exposure, toxic chemicals, chronic inflammation, errors during cell division, even certain protective mechanisms designed to prevent cancer. When a cell experiences enough stress or damage, it may receive a signal. Stop dividing, stop reproducing, do not pass on damaged genetic material. From an evolutionary perspective, this is often a good thing. A severely damaged cell continuing to divide could become dangerous. It might contribute to cancer. It might spread genetic errors. The body therefore possesses safety mechanisms that can place cells into a kind of permanent retirement. The cell remains alive, but its reproductive career is over. At first glance, this seems beneficial, and in many situations it is. Cellular senescence helps protect us. It acts as a biological emergency brake. When something appears wrong, the cell stops. Problem solved. Or so scientists once thought. The trouble begins when these retired cells fail to leave. In a healthy young body, the immune system often identifies senescent cells and removes them. The cleanup process works efficiently. Damaged cells are cleared away. Healthy cells take their place. The system remains balanced. But as we age, that cleanup system becomes less effective. more senescent cells appear, fewer are removed. Gradually, they begin accumulating throughout tissues and organs. The abandoned factories start multiplying, and this is where the zombie cell problem truly begins. Because senescent cells are not silent. They do not simply sit harmlessly in the background. They actively communicate with surrounding cells. Unfortunately, much of that communication is bad news. Scientists discovered that senescent cells release a complex mixture of chemical signals, inflammatory molecules, growth factors, proteins, biological messengers. Collectively, these secretions are often referred to as the senescence-associated secretory phenotype. The name may sound complicated, but the idea is simple. Zombie cells become biological polluters. Instead of helping the body function properly, they begin broadcasting distress signals. Imagine that abandoned factory leaking toxic waste into the city's water supply. Nearby buildings suffer damage. Workers become sick. Productivity declines. Soon, the effects spread far beyond the original factory. This appears to be exactly what senescent cells do. Their inflammatory signals affect neighboring cells. Healthy cells become stressed. Tissue function deteriorates. Repair processes become less efficient. The local biological environment grows increasingly hostile. One damaged cell can influence many others, and over time the damage spreads. Researchers have observed senescent cells in numerous aging tissues: skin, muscles, blood vessels, joints, the immune system, the brain. Virtually every major organ system appears vulnerable to their accumulation. As the number of zombie cells increases, aging itself may begin accelerating, not because of one catastrophic failure, but because of countless small disruptions occurring simultaneously. This realization transformed how scientists think about aging. For many years, aging was viewed primarily as a process of wear and tear. Cells became damaged, tissues deteriorated, function gradually declined. That explanation still matters. But the zombie cell theory adds another layer. Some aspects of aging may not result solely from damage. They may result from damaged cells actively spreading dysfunction throughout the body. The difference is profound. Instead of passive decay, we are looking at active biological sabotage. And one of the most important consequences involves inflammation. Inflammation is a fascinating process. In the short term, it is essential for survival. When you cut your finger, inflammation helps coordinate repair. When pathogens invade, inflammation helps mobilize immune defenses. Without inflammation, healing would be impossible. The problem arises when inflammation becomes chronic, when it never fully shuts off, when the body remains trapped in a low-level state of biological alarm. Many aging researchers now believe that chronic inflammation is one of the defining features of growing older. Some even use the term inflammaging, a combination of inflammation and aging. The idea is simple. As zombie cells accumulate, they release inflammatory signals. Those signals contribute to chronic inflammation. The inflammation damages tissues. Damaged tissues generate more stressed cells. More stressed cells become senescent. More senescent cells produce more inflammation. A vicious cycle emerges. The process feeds itself, like a small fire slowly spreading through a forest. At first, the flames are barely noticeable, but over time, the entire landscape changes. This chronic inflammatory state has been linked to numerous age-related conditions: cardiovascular disease, osteoarthritis, metabolic disorders, neurodegenerative diseases, frailty, declining immune function. Scientists are still debating the exact role zombie cells play in each condition. But the evidence continues growing. Where senescent cells accumulate, problems often follow. The implications are extraordinary, because if zombie cells help drive aging, perhaps removing them could improve health. This question gave birth to an entirely new scientific field: senolytics. The name comes from senescence and lytic, meaning destruction. Senolytics are therapies designed to identify and eliminate senescent cells. In essence, they are biological cleanup crews. Their goal is straightforward: find the abandoned factories, remove them, allow healthy tissue to function more effectively. At first, the idea sounded almost too simple. Could removing a relatively small number of dysfunctional cells really influence aging? Animal studies produced surprising results. In some experiments, researchers genetically removed senescent cells from aging mice. The outcomes attracted enormous attention. The animals often demonstrated improvements in multiple measures of health. Some showed better physical function, Others exhibited healthier tissues. Certain age-related problems appeared reduced. These findings generated tremendous excitement. For the first time, scientists had evidence suggesting that eliminating specific aging cells might improve overall health. Importantly, the goal was not immortality. The goal was healthier aging, a longer health span, more years spent functioning well, more years free from disease and disability. Today, researchers around the world are investigating various senolytic approaches. Some involve pharmaceutical compounds. Others explore immune-based strategies. Some scientists hope to train the immune system to recognize and remove senescent cells more effectively. Others seek drugs capable of targeting zombie cells directly. The field remains young. Many questions remain unanswered. Researchers must determine which senescent cells are harmful, which may still serve useful functions. How often treatment should be administered? What side effects might emerge? Biology rarely offers simple solutions. Every intervention carries risks. Every system interacts with countless others. Yet the promise remains compelling. Because senolytics represent something larger than a new medical treatment. They represent a new way of thinking about aging itself. For centuries, aging was viewed as inevitable decline, something to endure rather than understand. Now scientists are beginning to identify specific mechanisms contributing to the process. Telomere shortening, DNA damage, cellular dysfunction, and now zombie cells. Each discovery reveals another piece of the puzzle. Each discovery suggests that aging is not one thing. It is many things. Multiple processes unfolding simultaneously, multiple biological clocks ticking together, and perhaps most importantly, some of those processes may be modifiable. This does not mean aging will disappear. It does not mean humans will live forever. But it does mean that science is gradually transforming aging from an abstract mystery into a collection of understandable biological mechanisms. The more we understand those mechanisms, the greater our ability to influence them. Still, even if zombie cells vanished tomorrow, another challenge would remain, because every living cell depends on energy. Every heartbeat, every thought, every movement, every repair process, every immune response, all require power. And deep inside nearly every cell are tiny structures responsible for generating that power. Structures so important that life as we know it would be impossible without them. Yet these microscopic power stations do not remain young forever. As the years pass, they begin to falter. Their efficiency declines, their output weakens. The energy grid of the body slowly starts breaking down. And when that happens, the consequences can be felt everywhere. Because even healthy cells face another problem. Their power stations slowly begin to fail. Nasher Part 5, the Mitochondria Crisis, when the power grid starts collapsing. Imagine waking up in a city that never sleeps. Millions of lights illuminate the streets, hospitals operate around the clock, factories manufacture essential goods, transportation systems move people across vast distances, water treatment plants function continuously, communication networks remain active every second of every day. Everything depends on one thing: electricity. The moment the power begins failing, the entire city starts struggling. Traffic lights stop working, factories slow down, communication systems become unreliable, hospitals switch to emergency generators. What was once a thriving metropolis gradually becomes less efficient, less reliable, less resilient. Now imagine that this city is your body. Every heartbeat, every breath, every thought, every movement, every repair process, every immune response, every cellular activity requires energy. And hidden inside nearly every cell are microscopic power stations responsible for generating that energy. These remarkable structures are called mitochondria. If telomeres are among the body's biological clocks, mitochondria are its power plants. And many scientists believe that one of the most important drivers of aging is the gradual decline of these cellular energy systems. To understand why, we must begin with a remarkable story. A story that stretches back billions of years, long before humans existed, long before dinosaurs, long before plants covered the land, at a time when life itself was still young. One of the most fascinating theories in biology suggests that mitochondria were once independent living organisms. Billions of years ago, ancient single-celled organisms dominated Earth. At some point, one primitive cell appears to have absorbed another. Normally, the engulfed cell would have been destroyed. But something unusual happened. Instead of dying, the smaller organism survived inside its host. The two formed a partnership. The smaller organism provided energy. The larger organism provided protection and resources. Over immense spans of time, they evolved together. Eventually, they became inseparable. The smaller organism transformed into what we now call mitochondria. The host evolved into the ancestors of complex life. In a sense, every human being carries the descendants of this ancient partnership inside their cells. The evidence supporting this idea is extraordinary. Mitochondria possess their own DNA, separate from the DNA found inside the cell nucleus. They reproduce independently, and they retain characteristics suggesting a very ancient origin. Every time you move your hand, every time your heart beats, every time you remember a name, you are relying on biological machinery whose origins may stretch back billions of years. The partnership has been astonishingly successful. Without mitochondria, complex life as we know it would almost certainly be impossible. But what exactly do mitochondria do? The answer is surprisingly simple. They generate usable energy. Your body constantly extracts energy from food, carbohydrates, fats, proteins. These nutrients contain stored chemical energy. However, cells cannot use that energy directly. It must first be converted into a form the body understands, a kind of biological currency. That currency is called ATP, adenosine triphosphate. The name sounds complicated, but the concept is straightforward. Think of ATP as the body's energy money. Just as a city uses currency to pay workers and purchase resources, cells use ATP to power biological activities. Every movement of a muscle requires ATP. Every nerve signal requires ATP. Every cellular repair process requires ATP. Every heartbeat requires ATP. Without ATP, life stops almost immediately. Mitochondria are the factories that produce this currency. They take nutrients and oxygen. Through an extraordinarily sophisticated series of chemical reactions, they convert those inputs into ATP. The process is remarkably efficient. A single cell may contain hundreds or even thousands of mitochondria, working continuously, producing energy every second. At this very moment, trillions upon trillions of ATP molecules are being generated throughout your body. The scale is almost impossible to imagine. Yet, despite their importance, mitochondria are not immune to aging. Like every other component of the body, they experience wear and tear, and as they age, problems begin to emerge. Imagine a city whose power plants gradually become less efficient. The facilities still operate. Electricity is still produced, but output declines. Maintenance becomes more difficult. Equipment begins breaking down. Power shortages become increasingly common. Something similar appears to happen inside aging cells. Young mitochondria are highly effective and energy producers. They generate large amounts of ATP while maintaining relatively stable operation. As the years pass, however, mitochondrial performance often declines. Energy production becomes less efficient. Damage accumulates. Communication between mitochondria becomes impaired. The entire cellular energy network begins struggling. Scientists refer to this process as mitochondrial dysfunction, and it may be one of the central hallmarks of aging. To understand why mitochondria age, we need to examine a surprising consequence of energy production itself. Because generating energy is not entirely clean. Every power plant produces waste. Every engine generates byproducts. Mitochondria are no exception. During ATP production, mitochondria create highly reactive molecules known as free radicals. The name sounds dramatic, and in some ways, it is. Free radicals are unstable molecules missing an electron. Because they are unstable, they aggressively seek electrons from nearby molecules. In the process, they can damage cellular structures. Proteins, lipids, DNA, cell membranes. Virtually any component of the cell can become a target. Now, it is important not to think of free radicals as purely evil. They are natural byproducts of life. In controlled amounts, they even serve useful biological functions. The problem arises when free radical production exceeds the body's ability to manage them. Imagine sparks flying from a fireplace. A few sparks are harmless. Thousands of sparks landing on dry wood create danger. The body possesses sophisticated antioxidant systems designed to neutralize free radicals. These defenses function continuously, identifying unstable molecules, stabilizing them, preventing excessive damage. But over time, the balance can shift. More free radicals, less effective defenses, greater accumulation of damage. This condition is known as oxidative stress. Oxidative stress has become one of the most studied concepts in aging research. Think of it as a slow form of biological rust. Just as metal exposed to oxygen gradually corrodes, cellular components exposed to oxidative stress gradually accumulate damage. The process is not dramatic. It does not happen overnight. Instead, it unfolds slowly. Year after year, decade after decade. A protein becomes slightly damaged. A membrane becomes slightly less efficient. A strand of DNA experiences a minor alteration. Individually, these events seem insignificant. Collectively, they begin changing the behavior of cells. And mitochondria are particularly vulnerable because they operate at the center of energy production. The very process that keeps them functioning also exposes them to risk. It is one of biology's great paradoxes. The machinery that sustains life may also contribute to aging. As mitochondrial damage accumulates, ATP production often declines. Cells receive less energy. Repair systems become less effective. Recovery slows. Resilience decreases. The consequences can be felt throughout the body. Consider one of the most common experiences associated with aging: fatigue. Many older adults describe a gradual decline in energy. Tasks that once felt effortless require greater effort. Recovery after physical activity takes longer. Stamina decreases. The reasons are complex and involve many systems, but mitochondrial aging appears to play a significant role. When cellular power plants produce less energy, the effects ripple outward. Muscles may perform less efficiently. The brain may function less optimally. Repair processes may slow. The entire organism feels the impact. To visualize this process, return to the city analogy. Imagine a large city experiencing rolling blackouts. The power has not failed completely. Lights still work in some areas. Hospitals remain operational. Transportation continues, but interruptions become increasingly common. Some neighborhoods lose electricity temporarily. Factories reduce production. Infrastructure struggles to keep up. The city remains alive, yet everyone notices something has changed. Efficiency declines. Productivity decreases. Maintenance falls behind. The city becomes more vulnerable. That vulnerability mirrors what happens inside aging tissues. Cells still function, organs still work, life continues, but the energy margin becomes smaller. The reserve capacity shrinks. Challenges that once seemed minor become more difficult to overcome. An infection, an injury, a period of stress. The aging body often recovers more slowly because the cellular energy network is operating under greater strain. This helps explain why aging rarely results from a single failure. Instead, it emerges from multiple interconnected processes. Telomeres shorten. Zombie cells accumulate. Mitochondria lose efficiency. Each process influences the others. Less energy means weaker repair systems. Weaker repair systems allow more damage to accumulate. More damage creates additional stress on cells. The cycle continues. Yet despite these challenges, mitochondrial research remains one of the most promising areas in longevity science. Researchers are investigating exercise, nutrition, fasting, novel pharmaceuticals, cellular signaling pathways, various interventions that may help support mitochondrial function. The goal is not simply to extend lifespan. The goal is to preserve vitality, to maintain the body's ability to generate energy efficiently for as long as possible. Because ultimately, aging is not merely about surviving. It is about functioning, moving, thinking, creating, experiencing life fully, and energy lies at the heart of all those abilities. Still, mitochondria are only one part of the story. Even if every cellular power plant operated perfectly, another threat would remain. A threat hidden within the very blueprint of life itself. Every cell depends on instructions, precise biological information that tells it how to function, how to repair itself, how to reproduce, how to survive. Those instructions are stored in DNA, the genetic instruction manual carried inside nearly every cell. For decades, scientists assumed DNA was relatively stable, a secure archive preserving life's most important information. But modern research has revealed a troubling reality. That instruction manual is constantly under attack. Every day, every hour, every minute. And as the years pass, the errors begin accumulating. Because as the energy supply weakens, something even more dangerous begins happening. The instruction manual itself starts accumulating errors. Part 6. DNA under attack every single day. Imagine owning the most valuable book in existence, a book so important that every function of an entire civilization depends upon it. Inside its pages are the instructions for building cities, maintaining infrastructure, producing food, generating power, educating future generations. Everything depends on the accuracy of that book. Now imagine that every day, countless threats attack it. Pages are torn, words are smudged, letters disappear, entire sentences become distorted. Fortunately, an army of librarians works around the clock repairing the damage. Most mistakes are corrected before anyone notices. The book survives. The civilization continues. But what happens if the damage begins accumulating faster than repairs can be made? What happens when tiny errors start slipping through? And what happens when those errors continue building for decades? In many ways, this is the story of aging. Because deep inside nearly every cell of your body exists something remarkably similar to that priceless instruction manual. DNA, the genetic blueprint of life, the archive containing the information necessary to build, maintain, and repair your body. When most people think of DNA, they imagine something stable, permanent, almost sacred, a flawless code quietly preserved inside the nucleus of every cell. The reality is far more dramatic. Your DNA is under attack every single day. Right now, as you listen, damage is occurring, not because something has gone wrong, but because damage is an unavoidable consequence of being alive. And understanding this fact may help explain one of the deepest causes of aging. Scientists use a term called genomic instability. It sounds complicated, but the idea is surprisingly simple. The genome is the complete collection of genetic information stored inside your cells. Genomic instability refers to the gradual accumulation of damage, errors, and disruptions within that information. Imagine a vast library containing millions of critical documents. If every day a few pages become damaged, misplaced, or altered, eventually the integrity of the library begins to suffer. Something similar happens to DNA throughout life. The remarkable question is not whether damage occurs. The remarkable question is how life survives despite it. Because DNA faces threats from every direction. Some originate outside the body, others originate from within. Let's begin with one of the most familiar: sunlight. Most people associate sunlight with warmth, nature, health, yet hidden within sunlight is ultraviolet radiation. These high-energy rays possess enough power to damage DNA directly. Every time sunlight reaches your skin, tiny molecular changes can occur. Your cells immediately begin responding. Repair crews are deployed. Damage is corrected. Most of the time, the system works astonishingly well, but not always. The occasional error remains. Over years and decades, those surviving errors accumulate. This is one reason excessive sun exposure is strongly associated with skin aging and skin cancer. The damage may be invisible at first, but the biological record remains. Then there is pollution. Modern environments contain countless substances our ancestors never encountered. Industrial emissions, vehicle exhaust, airborne particles, chemical contaminants. Many pollutants can generate oxidative stress inside cells. Others interact directly with DNA. Again, the body fights back. Protective systems activate, repair mechanisms engage. But every battle leaves the possibility of damage behind. Smoking provides another powerful example. Scientists have identified thousands of chemical compounds in cigarette smoke. Many possess the ability to damage DNA directly. Others increase inflammation. Others generate free radicals. Together, they create an environment that places enormous stress on cellular systems. Over time, the effects become measurable. The risk of cancer rises. Tissue function declines. Biological aging accelerates. The lesson is not merely that smoking is harmful. The lesson is that DNA exists in a constant struggle against environmental threats. Yet some of the most important sources of DNA damage come from inside the body itself. And this is where the story becomes truly fascinating. Remember the mitochondria from the previous chapter, the cellular power plants responsible for generating ATP. As they produce energy, they also generate free radicals. These reactive molecules are natural byproducts of metabolism. In other words, simply being alive creates risk. Every heartbeat, every breath, every calorie transformed into energy, all contribute in small ways to molecular wear and tear. Life damages itself while sustaining itself. At first, this seems almost paradoxical. How can the processes keeping us alive also contribute to aging? Yet biology is full of trade-offs. The very chemistry that makes complex life possible also creates opportunities for damage. Imagine driving a car. The engine allows movement, freedom, transportation. But every mile adds wear to the machine. Life operates similarly. The metabolic engine sustaining us also generates biological stress. This means DNA is not attacked occasionally, it is attacked continuously. Scientists estimate that each cell experiences thousands of DNA damage events every day. The number is astonishing. Thousands. Every single day. And yet most of us continue living for decades. How is that possible? The answer lies in one of the greatest achievements of evolution: DNA repair systems. If DNA is the instruction manual of life, then repair enzymes are its maintenance teams. Tiny molecular specialists patrol the genome constantly, searching for errors, identifying damage, replacing incorrect sections, restoring damaged structures. The sophistication of these systems is extraordinary. Some enzymes specialize in detecting broken DNA strands. Others remove damaged chemical groups. Others compare genetic information against backup templates and restore missing sections. Entire networks of molecular machinery cooperate to preserve genomic integrity. Without these repair systems, complex life would be impossible. Every day, countless errors are corrected before they can cause problems. Most of the damage occurring inside your body right now will never affect you. It will be identified, repaired, resolved, and forgotten. The process is so effective that we rarely think about it. But therein lies the challenge. Repair systems are not perfect. No repair system is perfect. Imagine a city repairing roads. If 1,000 potholes appear each day and crews fix 999 of them, the city functions remarkably well. Yet one pothole remains. The next day another remains. Then another. Eventually, even a highly effective maintenance system begins accumulating unfinished work. Something similar happens inside aging cells. For many years, repair keeps pace with damage. Then gradually, the balance shifts. Damage increases. Repair efficiency declines. Resources become limited. Errors accumulate faster than they can be corrected. Why does this happen? Partly because repair systems themselves are made from biological components. The repair crews age too. Proteins become damaged. Cellular energy declines. Communication systems weaken. Everything that maintains DNA is itself subject to aging. This creates a profound challenge. The systems responsible for preserving information slowly lose their ability to preserve information perfectly. The consequences emerge gradually. A single DNA error may be harmless. Many errors remain silent. Some occur in regions that do not significantly affect cellular function. Others are corrected during future repair cycles. But occasionally, an important mutation appears. A mutation is simply a change in genetic information. Sometimes mutations are beneficial, sometimes neutral, sometimes harmful. Over a lifetime, mutations accumulate throughout tissues. Most remain insignificant. Some alter cellular behavior. A few become dangerous. The connection between mutations and aging is complex. Scientists do not believe mutations alone explain aging. However, they clearly contribute. As errors accumulate, cells may function less efficiently. Communication may become disrupted. Repair mechanisms may become compromised. The probability of dysfunction gradually rises. It is similar to repeatedly making small mistakes while copying a massive encyclopedia. One error may not matter. Thousands eventually change the quality of the information. And nowhere is this more evident than cancer. Cancer and aging are deeply connected. The risk of many cancers rises dramatically with age. This relationship puzzled scientists for generations. Today, the explanation appears increasingly clear. Cancer often emerges from the accumulation of genetic changes. Not one mutation, not two, but many. The process usually takes years, sometimes decades. As DNA damage accumulates and repair systems miss occasional errors, the probability of dangerous cellular behavior increases. Eventually, a cell may acquire the wrong combination of mutations. The safeguards fail. Growth becomes uncontrolled. Cancer begins. This relationship reveals one of biology's most difficult balancing acts. The body must preserve cellular function for decades while simultaneously preventing uncontrolled growth. Too little repair leads to dysfunction. Too much cell division increases cancer risk. Too much cellular senescence may accelerate aging. Too little may permit damaged cells to survive. Nature constantly balances competing priorities. There is no perfect solution, only trade-offs refined through evolution. And perhaps this leads us to one of the most profound lessons hidden within aging. Life survives because it constantly repairs itself, not because it is flawless, not because it avoids damage, not because it is perfectly designed. Life survives because it is resilient, because it repairs, because it adapts, because it continues despite imperfection. Consider your own body. Every scar tells a story of repair. Every recovered illness reflects biological resilience. Every healed wound demonstrates the extraordinary capacity of living systems to restore themselves. For decades, perhaps even generations, your cells have fought an endless war against entropy, against disorder, against damage, against time. And for the most part, they have succeeded. The fact that you are alive at this moment is evidence of countless victories. Trillions upon trillions of successful repairs, yet perfection remains impossible. No repair system can achieve absolute accuracy forever. No biological machine can operate indefinitely without accumulating wear. No living organism can completely escape entropy. Aging in many ways may be the price complex life pays for existing at all. The gradual accumulation of imperfections, the slow emergence of errors, the inevitable limits of maintenance. And yet, as scientists investigated aging more deeply, they uncovered an intriguing mystery. DNA damage alone could not explain everything. Two cells with identical DNA could behave very differently. Two identical twins could age at different rates despite sharing nearly the same genetic blueprint. Something else appeared to be happening, something beyond the hardware itself. Imagine two computers built from identical components. One performs beautifully, the other struggles. The difference is not necessarily the hardware. It may be the software, the instructions controlling how the hardware operates, the programs determining which functions are active and which remain silent. And this realization opened one of the most revolutionary chapters in aging science. Because if DNA is the hardware of life, perhaps aging is not only about damaged hardware, perhaps the software is changing too. Number part 7, Epigenetics, the software that controls your destiny. Imagine that you are handed the most sophisticated computer ever created. Its hardware is flawless. The processors are powerful, the memory is vast, the design is extraordinary. But there is one problem. The software controlling the machine becomes confused. Important programs fail to launch. Critical instructions are ignored. Useful functions are switched off. Unnecessary processes run constantly. The hardware remains largely intact, yet the system performs poorly. Now imagine something similar happening inside the human body. For decades, scientists believed aging was primarily a story of physical damage. Telomere shortening, DNA accumulating errors, mitochondria losing efficiency, zombie cells spreading inflammation. All of these processes are important. But researchers eventually encountered a mystery. Something did not quite fit. People with similar genes often aged very differently. Even identical twins who share nearly the same DNA sometimes develop dramatically different health outcomes. One twin remained vibrant and healthy. The other developed disease earlier. One appeared biologically younger. The other aged more rapidly. If their genetic blueprint was nearly identical, what explained the difference? The answer led scientists into one of the most revolutionary discoveries in modern biology, a field known as epigenetics. And according to some researchers, epigenetics may hold one of the most important clues to understanding aging itself. To appreciate why, we must first understand a crucial distinction: the difference between genes and gene expression. Most people think of genes as destiny, a set of biological instructions inherited from our parents. And in some ways, that is true. Genes contain information. They help determine countless characteristics throughout life. But possessing a gene and using a gene are not the same thing. Imagine owning a library containing thousands of books. Simply having the books does not mean you are reading them. Some books remain closed. Others are consulted daily. Still others are opened only under specific circumstances. The same principle applies to genes. Every cell in your body contains essentially the same DNA. Yet brain cells behave differently from skin cells. Skin cells behave differently from liver cells. Liver cells behave differently from muscle cells. Why? Because different genes are activated in different cells. The DNA may be the same. The instructions being used are not. This process is called gene expression. Genes provide possibilities. Gene expression determines which possibilities become reality. And this brings us to epigenetics. The word literally means above genetics. It refers to a collection of mechanisms that regulate how genes are used without changing the underlying DNA sequence itself. Think of DNA as the script of a play. Epigenetics determines which actors appear on stage, which scenes are performed, which lines are emphasized. The script remains largely unchanged. The performance changes dramatically. Scientists often describe epigenetics as a control system layered on top of the genome. Chemical markers attach to DNA. Proteins interact with chromosomes. Complex molecular signals influence which genes are active and which remain silent. The result is a highly dynamic biological system capable of responding to the environment. And this is where the story becomes truly fascinating. Because unlike your DNA sequence, many epigenetic patterns can change throughout life. Your experiences help shape them. Your environment influences them. Your behavior affects them. In a very real sense, your biology is listening to how you live. Perhaps the best way to understand this concept is through a simple analogy. Imagine a grand piano, an enormous piano containing every note necessary to create beautiful music. The piano itself represents your DNA. The keys are the genes. Every person begins life with a particular instrument. Some pianos may have slight differences, just as people possess different genetic variations. But owning a piano does not automatically produce music. Someone must decide which keys to play, which notes to emphasize, which melodies to create. Epigenetics functions like the pianist. It determines which genetic keys are activated. Some genes become loud, others remain silent. Some are used frequently, others rarely at all. The music emerging from the instrument depends not only on the piano itself, but also on how it is played. This idea fundamentally changed how scientists view aging. Because if gene activity can change over time, then aging may involve more than accumulated damage. Perhaps the instructions controlling the body gradually become disrupted. Perhaps the biological software itself begins losing organization. Perhaps cells slowly forget how to function optimally. This possibility helps explain one of the most fascinating observations in longevity research: identical twins. At birth, identical twins possess nearly identical DNA. They begin life with remarkably similar genetic blueprints. In many ways, they are nature's perfect experiment. If genes alone determined aging, identical twins should remain highly similar throughout life. Yet reality tells a different story. Researchers studying twins have discovered that epigenetic differences accumulate over time, especially when twins live different lifestyles, different diets, different stress levels, different occupations, different environments, different sleep habits. As the years pass, their biological profiles begin diverging. The twins may still share similar DNA, but the activity of that DNA changes. One may develop certain diseases, the other may not. One may age more rapidly, the other may remain biologically younger. The difference is not necessarily written into the genes themselves. The difference may lie in how those genes are being regulated. This realization raises a profound question. What influences gene expression? The answer includes many of the factors we have already explored throughout this series: stress, food, exercise, sleep, environment, social connections. In other words, many aspects of daily life appear capable of interacting with the body's biological software. Consider stress. When humans experience stress, the body activates a complex network of hormonal responses. These systems evolved to help our ancestors survive danger. Short-term stress can be useful. The challenge arises when stress becomes chronic: months, years, decades. Persistent stress appears capable of influencing epigenetic patterns. The body receives signals suggesting that the environment is hostile. Certain genes become more active, others become less active. The biological consequences may accumulate over time. Nutrition appears capable of influencing gene expression as well. Food is not merely fuel. Food is information. Nutrients interact with cellular pathways. They provide signals that help regulate biological processes. Scientists continue exploring how different dietary patterns affect epigenetic regulation. The field remains complex, yet evidence increasingly suggests that nutrition influences far more than calorie intake alone. Exercise provides another powerful example. Physical activity does not simply strengthen muscles. It sends molecular signals throughout the body. Genes involved in repair, energy production, inflammation control, stress resistance. Many become more active following exercise. The body responds to movement by altering biological priorities. In a sense, exercise communicates instructions to your cells. Sleep may be equally important. Every night, the body enters an extraordinary state of restoration. Hormonal systems recalibrate, the brain clears waste products, repair processes intensify, gene activity shifts. When sleep becomes chronically disrupted, these carefully coordinated systems may become less organized. The effects extend far beyond feeling tired. They reach deep into the biology of aging itself. Taken together, these findings reveal something remarkable. The human body is not a passive machine following a fixed genetic script. It is an adaptive system, responsive, dynamic, continuously adjusting to its environment. Your genes matter, but so does how those genes are regulated. And perhaps no modern scientist has popularized this idea more than David Sinclair. Sinclair has become one of the most influential researchers in longevity science. Among his most discussed ideas is something known as the information theory of aging. The theory remains an active area of scientific debate, yet it offers a fascinating framework for thinking about growing older. According to this perspective, aging may not be driven solely by accumulated damage. Instead, aging may result partly from a loss of biological information. Imagine a compact disk containing valuable data. The information remains stored on the disk, but scratches make it increasingly difficult to read correctly. The data has not disappeared. Accessing it becomes more challenging. Sinclair suggests something similar may occur in aging cells. The genetic information remains largely intact, yet the epigenetic systems responsible for organizing and interpreting that information become disrupted. Cells gradually lose their ability to read the instructions properly. Over time, they forget aspects of their biological identity. A muscle cell becomes less effective at behaving like a youthful muscle cell. A skin cell becomes less effective at behaving like a youthful skin cell. The hardware remains present. The software becomes increasingly disorganized. Whether every aspect of this theory ultimately proves correct remains to be seen. Science is still investigating, but the implications are extraordinary. Because if aging involves information loss rather than irreversible destruction, then a tantalizing possibility emerges. Perhaps some aspects of aging can be reversed, not by rebuilding every damaged molecule, not by replacing every cell, but by restoring lost biological instructions, by helping cells remember who they are. This idea once sounded like science fiction. Today, it sits at the forefront of some of the world's most advanced aging research. Researchers are asking questions that would have seemed impossible only a generation ago. Can cellular identity be restored? Can aging tissues be rejuvenated? Can biological software be reset? The answers remain uncertain, yet the evidence emerging from laboratories around the world continues attracting enormous attention. Because for the first time, scientists are not merely studying how aging occurs, they are beginning to explore whether aspects of aging can be reversed. And that possibility leads directly to one of the most astonishing questions in modern biology. If the software becomes corrupted, can it ever be reset? Part 8. The cellular cleanup system we keep ignoring. Imagine living in a house for decades. Every day, objects enter the house. Furniture, clothes, books, boxes, appliances, tools. At first, everything is organized. The rooms feel spacious. The hallways are clear. The house functions beautifully. But now imagine something unusual. Nothing is ever thrown away. Broken furniture remains in the living room. Old newspapers pile up in the corners. Damaged appliances sit untouched. Boxes accumulate in every hallway. Year after year, clutter grows. Eventually, moving through the house becomes difficult. Repairs become harder. Finding useful items takes longer. The house still exists, but it functions less efficiently. Now imagine the opposite. Imagine a house with an exceptional cleanup crew. Old items are removed. Broken objects are recycled. Useful materials are recovered. Waste never accumulates for long. The house remains functional because maintenance never stops. In many ways, your body faces the same challenge. Every day, proteins become damaged. Cellular components wear out. Mitochondria lose efficiency. Molecular structures break down. Life continuously generates biological clutter. And if that clutter were allowed to accumulate indefinitely, cells would quickly become dysfunctional. Fortunately, evolution developed an extraordinary solution: a process so important that many scientists now consider it one of the most fundamental mechanisms of longevity, a process known as autophagy. The name comes from ancient Greek words meaning self-eating. At first, that sounds alarming. Why would cells eat themselves? The answer reveals one of the most elegant systems in all of biology. Autophagy is not self-destruction. It is self-renewal. It is the cellular equivalent of cleaning, recycling, and renovation, a sophisticated maintenance program operating inside your body every day. Imagine a city with an advanced recycling system. Old buildings are dismantled. Useful materials are recovered. Damaged equipment is replaced. Waste is removed before it causes problems. The city remains healthy because it continuously renews itself. Autophagy performs a remarkably similar function inside cells. Damaged proteins are identified. Worn-out cellular machinery is broken down. Defective mitochondria are removed. Materials are recycled. Useful components are reused. The process reduces clutter while simultaneously providing raw materials for repair and renewal. Without autophagy, cells would gradually fill with biological junk. The consequences would be devastating. Yet for many years, scientists only partially understood how this process worked. Then came a breakthrough that transformed the field. The story centers on a Japanese scientist named Yoshinori Ohsumi. During the late 20th century, Ohsumi became fascinated by a biological process that many researchers had largely overlooked. Using simple organisms known as yeast, he began investigating how cells dismantle and recycle their internal components. At the time, autophagy remained poorly understood. Scientists knew it existed, but many of its mechanisms remained mysterious. Ohsumi's research helped reveal the molecular machinery responsible for cellular recycling. His discoveries opened an entirely new window into how cells maintain themselves. The impact was enormous. Researchers suddenly gained a much deeper understanding of one of life's most essential maintenance systems. For these contributions, Ohsumi was awarded the Nobel Prize in Physiology or Medicine 2016. The award reflected something profound. Autophagy was no longer viewed as a minor biological curiosity. It had become recognized as a fundamental process supporting health and survival. To appreciate its importance, imagine what happens inside your body every day. Proteins are constantly being built, but proteins do not last forever. Eventually, they become damaged, misfolded, less efficient. Some may even become harmful. Similarly, mitochondria age. As we discussed in the previous chapter, mitochondrial dysfunction contributes to declining energy production. Old mitochondria often generate more oxidative stress. They become less effective power plants. If they remain in place indefinitely, they may damage surrounding cellular structures. Autophagy helps solve this problem. Cells identify dysfunctional components, encapsulate them, break them down, recover useful materials, and create space for healthier replacements. It is a remarkably intelligent system. Not because the cell thinks consciously, but because evolution has refined these processes over billions of years. The result is something that appears almost purposeful, a continuous cycle of destruction and renewal. A biological reminder that survival often depends not on preserving everything, but on knowing what to let go. This idea may help explain one of the great mysteries of aging. Why does aging occur at all if cells possess such sophisticated repair systems? Part of the answer appears to involve accumulation. Over time, waste begins outpacing removal. Damaged proteins accumulate. Defective mitochondria persist. Molecular debris builds up. The cleanup crews struggle to keep pace. Imagine a city generating more garbage than its sanitation department can remove. At first, the difference seems insignificant. A little extra waste here, a little extra waste there. But over years, the imbalance grows. Soon the streets begin filling with debris. Infrastructure suffers. Disease spreads. Efficiency declines. Something similar appears to occur during aging. Autophagy continues operating, but often less efficiently. Cellular clutter accumulates. The biological environment becomes increasingly crowded and dysfunctional. Scientists now believe that declining autophagy may contribute to numerous age-related conditions: neurodegenerative diseases, metabolic disorders, cardiovascular problems, immune dysfunction. The exact mechanisms remain under investigation, yet the overall pattern is becoming increasingly clear. Healthy maintenance matters, and maintenance depends upon cleanup. This realization led researchers to an intriguing question. Can autophagy be stimulated? Can the body's recycling systems be encouraged to work more effectively? The answer appears to be yes, at least under certain conditions. And one of the most fascinating triggers involves something humans have experienced throughout history: not eating, or more precisely, periods without food. For most of human existence, constant access to food was impossible. Our ancestors frequently experienced periods of scarcity. Meals were not available every few hours. There were times of abundance and times of fasting. The human body evolved within that environment. When food became scarce, survival required adaptation. One adaptation appears to involve increased cellular recycling. Think about it from the perspective of the cell. If new resources are not arriving, existing resources become more valuable. Waste becomes potential fuel. Old components become reusable building materials. The body shifts into a conservation and renewal mode. Autophagy increases. Cells begin cleaning house. Damaged proteins are removed, defective structures are recycled. The system becomes more active. This connection between fasting and autophagy has generated enormous scientific interest. Researchers continue investigating how different fasting patterns influence cellular maintenance. The field remains active and evolving. Not every question has been answered. Not every claim circulating online is supported by strong evidence. Yet one theme continues appearing throughout the research. Periods without constant nutrient intake appear capable of activating important cellular stress response pathways. These pathways often overlap with mechanisms associated with longevity. This brings us to a topic many listeners may already recognize: intermittent fasting. In a previous exploration, we discussed how intermittent fasting may influence metabolism, insulin sensitivity, and cellular repair. Autophagy provides one possible biological explanation for some of those observations. When the body spends time in a fasting state, certain repair and maintenance systems appear to become more active. The body shifts priorities. Growth temporarily slows. Maintenance increases. Cleanup accelerates. Again, this does not mean fasting is a magical cure. It does not mean everyone should follow the same eating pattern. Human biology is complex. Individual needs vary. Medical conditions matter. Scientific nuance matters. But the broader lesson is fascinating. The body may benefit from periods devoted not only to growth and consumption, but also to repair and renewal. And this idea raises an uncomfortable question about modern life. Because unlike our ancestors, many people today rarely experience meaningful breaks from food: breakfast, snacks, coffee drinks, lunch, afternoon snacks, dinner, dessert, late-night eating. The body often remains in a nearly continuous fed state. For many individuals, nutrient intake extends across most waking hours. From a historical perspective, this pattern is unusual. The human body evolved under very different conditions. Scientists are increasingly exploring whether constant eating may interfere with certain maintenance processes. If cellular cleanup systems become most active during periods of nutrient scarcity, then reducing those periods could theoretically reduce opportunities for repair. Researchers continue debating the extent of these effects. Much remains to be learned, yet the question itself is revealing. Modern life has dramatically altered the biological environment in which humans evolved. Artificial lighting extends the day. Technology reduces physical activity. Food is available almost constantly. Our biology developed under one set of conditions. We now live under another. Understanding that mismatch may be essential for understanding aging. Because aging is not merely the result of time passing. It is the result of countless interactions between our biology and our environment. And increasingly, scientists suspect that some aspects of modern life may be accelerating the process. We have explored telomeres, zombie cells, mitochondria, DNA damage, epigenetic changes. Autophagy now joins that growing list. Another mechanism influencing how we age. Another reminder that health depends not only on building and growing, but also on cleaning and renewing. The body is not a machine that thrives through constant activity alone. It is a system that requires cycles: activity and recovery, growth and maintenance, consumption and renewal. When those cycles become disrupted, problems begin accumulating. And that brings us to a sobering realization. Modern life may not merely slow the body's cleanup systems. It may be creating entirely new pressures that accelerate aging itself. Because modern life doesn't merely slow cellular cleanup, it may actively accelerate aging itself. Part 9. The modern world is aging us faster. Imagine taking a healthy human being from 200 years ago and placing them into the modern world. They wake up under artificial light. They spend most of the day sitting. Food is available every hour. Stress arrives through glowing screens. Sleep is shortened. The air contains pollutants their ancestors never encountered. Their social interactions increasingly occur through technology rather than face-to-face connection. Would their body thrive? Or would it struggle to adapt? This question sits at the heart of one of the most important debates in aging science. For most of human history, aging was viewed as a simple consequence of time. The years passed, the body deteriorated, the process seemed inevitable. But modern research is revealing something far more complicated. Time matters. Genetics matter. Biology matters. Yet the environment matters too. And the environment humans now inhabit may be unlike anything our species has ever experienced. Many scientists are beginning to ask a provocative question. Have humans created the fastest aging environment in history? The answer remains uncertain, but the evidence supporting the question continues to grow. Because while aging has always existed, modern life may be amplifying many of the biological processes that drive it. One of the clearest examples begins with something sweet, sugar. Few substances are as deeply woven into modern diets. It appears in beverages, snacks, breakfast foods, desserts, sauces, processed meals. Even foods that do not taste particularly sweet often contain surprising amounts of added sugar. The human brain evolved to appreciate sweetness. For our ancestors, sweet foods often signaled valuable energy sources. Fruit, honey, seasonal abundance. The challenge is that modern food systems provide sweetness at a scale evolution never anticipated. And excessive sugar appears capable of influencing aging through several pathways. One of the most fascinating is a process called glycation. Imagine dripping syrup onto a complex machine. At first, the syrup seems harmless, but over time it begins sticking to gears, coating moving parts, interfering with normal function. Something similar can occur inside the body. Excess sugar molecules can attach to proteins and other biological structures. These unwanted attachments create compounds known as advanced glycation end products. Scientists often shorten the name to AGEs. The acronym is fitting, because these compounds are closely associated with aging. As AGEs accumulate, tissues may become less flexible. Proteins may function less efficiently. Inflammation may increase. Damage gradually builds. The effects are particularly visible in structures that rely on elasticity: skin, blood vessels, connective tissue. Over time, glycation may contribute to wrinkles, vascular stiffness, and other signs commonly associated with aging. The process is not dramatic. It unfolds quietly, year after year, meal after meal, a slow biological consequence of chronic excess. Yet sugar is only one piece of the puzzle. Another powerful force shaping modern biology is stress. Not the brief stress our ancestors experienced when escaping danger, not the short bursts that help the body respond to challenges. Those forms of stress are normal, sometimes even beneficial. The problem is chronic stress, the kind that never fully disappears. Financial worries, work pressure, information overload, constant notifications, social comparison, uncertainty about the future. Many people live in a near-continuous state of psychological tension. The body interprets this tension through ancient biological systems. Chief among them is a hormone called cortisol. Cortisol is often called the stress hormone. In reality, its functions are far more complex. It helps regulate energy, immune activity, blood sugar, inflammation, circadian rhythms. Under normal conditions, cortisol plays an essential role in survival. The trouble begins when cortisol remains elevated for extended periods. Imagine a city operating under permanent emergency conditions. Sirens never stop. Emergency crews remain constantly activated. Resources are continuously redirected toward immediate threats. At first, the city functions. Eventually, exhaustion sets in. Maintenance suffers. Infrastructure deteriorates. Long-term planning disappears. Something similar may happen inside the body. Chronic cortisol elevation has been associated with inflammation, metabolic dysfunction, immune disruption, accelerated biological aging. Even telomere shortening appears linked to prolonged psychological stress in some studies. The body is listening to the mind more closely than many people realize. And in today's world, that conversation rarely pauses. The challenge becomes even greater when stress collides with another modern epidemic: Sleep deprivation. For most of human history, darkness dictated rest. The sun set, activity slowed, the body followed natural rhythms. Today, artificial lighting has transformed the relationship between humans and night. Work continues, entertainment continues, scrolling continues, notifications continue. The biological night often arrives long after the clock says bedtime. Many people accept inadequate sleep as normal, an inconvenience, a necessary sacrifice. Yet sleep is not merely rest. Sleep is biological maintenance. It is when the brain clears waste, hormones recalibrate, memories consolidate, repair systems activate, immune function strengthens, cells perform critical housekeeping tasks. When sleep becomes chronically restricted, the consequences ripple throughout the body. Inflammation rises, metabolic health suffers, cognitive performance declines, stress responses become exaggerated. The biological systems protecting long-term health begin falling behind. It is difficult to imagine building a healthy future while consistently stealing time from one of the body's most important recovery processes. Yet millions of people do exactly that every day. Then there is movement, or perhaps more accurately, the absence of movement. Human beings evolved in motion, walking, carrying, climbing, hunting, gathering, building. Physical activity was woven into survival itself. Today, much of modern life occurs while sitting. We sit while working. We sit while commuting. We sit while eating. We sit while relaxing. We sit while communicating. Entire days may pass with surprisingly little movement. The body, however, still expects activity. Muscles evolved to be used. Bones evolved to bear loads. Cardiovascular systems evolved to support movement. When physical activity disappears, biological consequences emerge. Muscle mass declines. Insulin sensitivity worsens. Mitochondrial function may suffer. Inflammation may increase. The body begins adapting to inactivity. Unfortunately, many of those adaptations move in the wrong direction. The phrase sedentary lifestyle sounds harmless, almost gentle. Yet, prolonged inactivity may be one of the most significant contributors to modern health decline. The human body was designed for motion. When motion disappears, health often follows. Food presents another challenge, not merely because of quantity, but because of quality. For most of human history, food came from recognizable sources: plants, animals, natural ingredients. Preparation required time and effort. Modern food systems have created something entirely new: ultra-processed foods. These products are often engineered for convenience, shelf stability, and intense flavor. They frequently contain combinations of refined sugars, industrial oils, additives, flavor enhancers, and highly processed ingredients rarely encountered in traditional diets. The issue is not that every processed food is inherently harmful. The issue is scale. In many countries, ultra-processed foods now represent a substantial portion of daily calorie intake. Researchers continue investigating the long-term consequences, yet associations with obesity, metabolic disease, inflammation, and poor health outcomes appear repeatedly throughout the scientific literature. The body evolved expecting one nutritional environment. It now receives another, and the biological adjustment may come at a cost. Meanwhile, invisible threats surround us. Environmental pollution. Air pollution alone exposes billions of people to microscopic particles capable of entering the body. These particles may contribute to inflammation, oxidative stress, cardiovascular disease, respiratory problems. Scientists are increasingly exploring connections between pollution exposure and biological aging markers. The evidence continues growing. Our ancestors faced many dangers, but widespread industrial pollution was not one of them. Modern biology must navigate challenges evolution never anticipated. Yet perhaps the most surprising aging factor of all is not physical. It is social. Human beings are profoundly social creatures. For hundreds of thousands of years, survival depended on connection. Families, tribes, communities, shared responsibility, shared protection, shared meaning. Isolation was often dangerous. Today, despite unprecedented digital connectivity, loneliness has become increasingly common. People may communicate constantly while feeling deeply disconnected. Social isolation is not merely an emotional experience. It has biological consequences. Research has linked chronic loneliness with increased inflammation, elevated stress responses, poorer health outcomes, even higher mortality risk. The body appears to interpret prolonged isolation as a threat, a signal that something is wrong. In many ways, social connection functions like nutrition or sleep. Or exercise, not a luxury, a biological necessity. When it is missing, health suffers. And when we step back and view the entire picture, a remarkable pattern emerges. Excess sugar, chronic stress, sleep deprivation, physical inactivity, ultra-processed foods, environmental pollution, social isolation. Each factor influences aging through different pathways, yet many converge on the same biological mechanisms: inflammation, oxidative stress, hormonal disruption, mitochondrial dysfunction, DNA damage, epigenetic alterations, the very processes we have explored throughout this series. Which brings us back to that provocative question: have humans created the fastest aging environment in history? Perhaps. Perhaps not. Science has not reached a final verdict, but one thing seems increasingly clear. Modern life often pushes biological systems in directions they were never designed to handle continuously. The result may be an acceleration of the aging processes already present within us. Yet there is good news hidden within this realization. If aspects of aging can be accelerated by lifestyle and environment, then they may also be influenced in the opposite direction. The same biology that responds to harmful signals can often respond to beneficial ones. The same systems that deteriorate under chronic stress can improve under supportive conditions. The same body that adapts to unhealthy environments can adapt to healthier ones. And this realization has inspired one of the most ambitious scientific efforts in human history. Researchers around the world are no longer asking only how aging happens. They are asking whether aging itself can be slowed, perhaps even partially reversed. What once belonged to the realm of mythology is becoming a serious scientific pursuit. And this is where the story becomes truly fascinating. Because if aging can be accelerated, perhaps it can also be slowed. And this is where modern science becomes truly fascinating. Part 10, the race to reverse aging. For thousands of years, humanity searched for immortality in all the wrong places. Ancient kings sent expeditions in search of mythical fountains. Alchemists attempted to transform ordinary substances into elixirs of eternal life. Emperors consumed strange potions hoping to defeat death. Legends promised magical waters, sacred relics, hidden secrets. Yet despite all the stories, aging remained undefeated. Wrinkles appeared. Hair turned gray. Bodies weakened. Generations came and went. Time always won. But something extraordinary has happened during the last few decades. For the first time in history, scientists are no longer approaching aging as a mystery beyond human understanding. They are treating it as a biological problem. A problem that can be studied, measured, influenced, perhaps one day even modified. This does not mean immortality is around the corner. Far from it. Many headlines exaggerate what modern science has achieved. Many claims spread faster than evidence. Yet beneath the hype, something remarkable is happening. Researchers around the world are identifying specific mechanisms that contribute to aging. And once a mechanism is understood, an obvious question follows. Can it be changed? Can it be repaired? Can it be slowed? Can it be reversed? These questions have launched one of the most ambitious scientific races in history, the race to reverse aging. One of the most promising areas involves the zombie cells we discussed earlier. Senescent cells, cells that have stopped functioning properly but refuse to disappear. Scientists discovered that these cells may contribute to inflammation, tissue dysfunction, and age-related decline. Naturally, researchers wondered whether removing them might improve health. This idea gave rise to senolytic drugs. Senolytics are compounds designed to identify and eliminate senescent cells while leaving healthy cells largely untouched. Think back to the abandoned factory analogy. Imagine a city filled with old industrial buildings leaking toxic waste into surrounding neighborhoods. One solution would be to remove those abandoned structures, clear the contamination, create space for healthier development. Senolytics aim to do something similar inside the body. Animal studies have produced encouraging results. In certain experiments, removing senescent cells improved physical function, reduced age-related decline, and in some cases, extended healthy lifespan. Researchers remain cautious. Human biology is vastly more complicated than laboratory animals. Many questions remain unanswered. Which senescent cells should be removed? How often? At what age? Could unintended side effects occur? The science is still evolving, yet the field continues advancing rapidly. Another major frontier involves stem cells. To understand why stem cells attract so much attention, Imagine a city's repair crews gradually disappearing. Road damage accumulates, buildings deteriorate, infrastructure becomes harder to maintain. Even small problems linger longer than before. Something similar happens in aging tissues. The body's regenerative capacity declines, repair slows, recovery becomes less efficient. Stem cells play a central role in this process. These remarkable cells possess the ability to develop into specialized cell types. They help replace damaged tissue, support healing, maintain biological function. In many tissues, stem cell activity decreases with age. Researchers hope that restoring or enhancing stem cell function may help rejuvenate aging organs. Numerous experimental therapies are now exploring this possibility. Some focus on stimulating existing stem cells, others investigate transplantation approaches. Still others seek to understand the molecular signals controlling regeneration. The promise is enormous, yet so are the challenges. Biology rarely offers shortcuts. The same cellular growth that promotes healing can sometimes increase cancer risk. Balancing regeneration and safety remains one of the field's greatest obstacles. But perhaps the most astonishing development in aging science emerged from an unexpected discovery. A discovery so surprising that it transformed how researchers think about cellular identity itself. The story begins with a Japanese scientist named Shinya Yamanaka. For many years, scientists assumed that once a cell became specialized, its identity was largely permanent. A skin cell was a skin cell, a muscle cell was a muscle cell, a nerve cell was a nerve cell. The process appeared irreversible. Then Yamanaka demonstrated something extraordinary. By introducing a small set of specific genes, mature cells could be reprogrammed into a more youthful stem cell-like state. The genes became known as the Yamanaka factors. The discovery stunned the scientific community. Cells appeared capable of forgetting aspects of their age and specialization. They could, in a sense, travel backward along the developmental timeline. The implications were profound. If cellular identity could be reset, what did that mean for aging? Could old cells become young again? The answer proved both exciting and complicated. Fully reprogramming cells can erase their specialized identity entirely. That may be useful in certain laboratory settings, but it is not practical for rejuvenating tissues inside a living person. A heart cell must remain a heart cell. A liver cell must remain a liver cell. Researchers therefore began exploring a more subtle approach: partial cellular reprogramming. Instead of completely resetting a cell, what if scientists could rewind only certain aspects of aging? Imagine restoring an old computer to an earlier version of its operating system without erasing all the files. Could cellular age be reduced while preserving cellular identity? Animal studies have produced intriguing results. Some experiments suggest that aspects of biological aging can indeed be partially reversed. Tissues appear healthier, function improves, biological markers become more youthful. The research remains in its early stages. Many technical hurdles remain, yet few areas of longevity science generate more excitement. Because if aging partly reflects lost cellular information, then restoring that information could fundamentally transform medicine. Another important area focuses on molecules called sirtuins. These proteins help regulate numerous cellular processes related to stress resistance, metabolism, DNA repair, and longevity. Interest in sirtuins increased after researchers observed that certain organisms activate survival pathways during periods of environmental challenge. Fasting, calorie restriction, nutrient scarcity. Under these conditions, cells often shift resources toward maintenance and repair. Sirtuins appear to play an important role in coordinating some of these responses. Scientists continue investigating how these pathways influence aging. Much remains unknown, yet the broader concept is fascinating. The body possesses ancient survival programs designed to protect cellular function during difficult times. Understanding those programs may reveal new strategies for extending health span. Closely related to this research is a molecule called NAD+, nicotinamide adenine dinucleotide. Like ATP, the name sounds intimidating, but its importance is easier to understand than its name suggests. NAD+ participates in countless cellular processes: energy production, DNA repair, metabolic regulation, stress responses. Many essential systems depend upon it. One observation has attracted particular attention. NAD+ levels often decline with age. As levels decrease, certain repair and maintenance pathways may function less effectively. Researchers are actively investigating whether restoring NAD+ levels could support healthier aging. The field has generated enormous interest. Numerous studies are underway. Some results appear promising. Others remain uncertain. As with much of longevity science, enthusiasm must be balanced with caution. The questions are often clearer than the answers. Yet the search continues. Then there is gene therapy, perhaps one of the most powerful technologies modern biology has ever developed. Gene therapy allows scientists to modify, replace, or supplement genetic information. Instead of treating symptoms, the goal is to address underlying biological causes. The potential applications are vast: inherited diseases, cellular repair, regenerative medicine, longevity interventions. Researchers are increasingly exploring how gene therapy might influence aging pathways. Could damaged repair systems be strengthened? Could protective genes be enhanced? Could cellular resilience be increased? These possibilities remain largely experimental, yet progress continues accelerating. What once sounded like science fiction is gradually entering the realm of scientific reality. And now another revolutionary force is entering the story: artificial intelligence. For most of human history, scientific discovery moved at the speed of human analysis. Researchers examined data, identified patterns, tested hypotheses. The process worked, but it was often slow. Modern biology generates enormous quantities of information: genetic data, protein interactions, cellular pathways, clinical results. The complexity is staggering. Artificial intelligence offers a new approach. Advanced systems can analyze vast data sets far more quickly than traditional methods. Patterns hidden within mountains of biological information may become visible. Potential drug candidates can be identified more efficiently. Complex relationships between genes, proteins, and diseases can be explored at unprecedented scale. Many researchers believe artificial intelligence may dramatically accelerate longevity research during the coming decades. Not because machines will solve aging alone, but because they may help scientists navigate complexity more effectively. The result is a remarkable convergence: biology, genetics, medicine, computing, artificial intelligence, regenerative science, all moving toward the same question: can healthy human lifespan be extended? Which leads us to one of the most fascinating discussions in the field. Could the first person to live to 150 already be alive? Some researchers believe it is possible, others remain skeptical. No one knows. The current scientific evidence does not support the idea that humans can routinely reach such ages today. Yet history offers a humbling lesson. Again and again, predictions about biological limits have proven incomplete. A century ago, many experts considered modern lifespans impossible. Advances in sanitation, vaccination, nutrition, medicine, public health, all transformed human longevity. The question is whether future breakthroughs could produce another transformation. Perhaps not immortality, perhaps not even 150 years, but something significantly beyond what we currently consider normal. The answer remains unwritten. Yet regardless of what the future holds, one truth often gets lost amid the excitement. Many people become fascinated by futuristic therapies while overlooking something important. The most powerful longevity tools are not hidden inside advanced laboratories. They are not waiting for regulatory approval. They are not decades away. Many already exist. And in the next chapter, we will explore a surprising reality. While scientists race toward the future, many of the most powerful anti-aging tools are already available today. Part 11: How to Slow Aging Starting Right Now. After everything we have explored in this journey, a natural question emerges. If aging is influenced by telomeres, mitochondrial decline, DNA damage, zombie cells, inflammation, epigenetic changes, and countless other biological processes, what can an ordinary person actually do? Most people will never have access to experimental gene therapies. Few will participate in cutting-edge longevity trials. The future technologies we discussed may take years, perhaps decades, to become widely available. So what about today? What about this week? What about the choices available right now? The answer is both encouraging and surprisingly so. Scientists can continue searching for revolutionary anti-aging treatments. Many of the strongest tools already exist. They are not glamorous. They do not generate dramatic headlines. They rarely go viral on social media. Yet again and again, scientific research points toward the same conclusion. The foundations of healthy aging are often remarkably ordinary: movement, sleep, nutrition, connection, purpose, stress management. Not because these things stop aging, nothing does, at least not yet, but because they influence nearly every biological system involved in aging. The challenge is that humans often search for miracles. We want the secret pill, the hidden shortcut, the revolutionary breakthrough, the single intervention that changes everything. Biology rarely works that way. The body is a network, thousands of systems interacting continuously. Improving one area helps. Improving several areas creates something much more powerful. And perhaps nowhere is this clearer than exercise. For decades, researchers have searched for medications capable of producing the benefits of physical activity. So far, nothing comes close. Exercise affects almost every major system in the body: cardiovascular health, metabolic function, brain health, immune activity, hormonal regulation, muscle maintenance, mitochondrial performance, inflammation control. The list seems endless. It is difficult to think of another intervention with such broad influence. This is why many scientists describe exercise as medicine, not because it cures everything, but because its effects are astonishingly widespread. Imagine designing a drug that improved energy production, reduced inflammation, strengthened bones, enhanced insulin sensitivity, improved mood, supported brain function, and lowered the risk of numerous chronic diseases. Such a drug would likely become one of the most valuable medical discoveries in history. Yet movement has been available to humans for hundreds of thousands of years. The difficulty is not access. The difficulty is consistency. Among the various forms of exercise, one has received increasing attention in longevity research: strength training. For much of modern history, cardiovascular exercise dominated health discussions. Walking, running, cycling, swimming, all remain incredibly valuable. But researchers have become increasingly interested in muscle itself, not merely as tissue that enables movement, but as an organ deeply connected to long-term health. As people age, muscle mass naturally declines, a process known as sarcopenia. The consequences extend far beyond physical appearance. Reduced muscle mass is associated with weakness, falls, loss of independence, metabolic dysfunction, increased vulnerability during illness. Strength training helps counter this decline. It provides a signal that muscle remains necessary, useful, worth maintaining. The body responds accordingly. Muscle is preserved, function improves, resilience increases. In many ways, strength training is not simply about building muscle. It is about preserving capability, preserving independence, preserving options, the ability to carry groceries, climb stairs, recover from injury, remain active later in life. Longevity is not merely about adding years. It is about protecting the quality of those years. Then there is sleep, the anti-aging intervention most people willingly sacrifice. Modern culture often treats sleep as negotiable, a luxury, something flexible. Yet biology disagrees. Sleep is when much of the body's maintenance occurs. The brain clears metabolic waste, hormonal systems recalibrate, immune processes strengthen, memory consolidates, cellular repair accelerates. Throughout this series, we have discussed DNA repair, inflammation, stress hormones, mitochondrial function. Many of these systems are influenced directly by sleep quality. When sleep suffers, everything else becomes harder. Stress becomes more difficult to manage. Food cravings increase. Exercise performance declines, recovery slows, decision-making worsens. It is difficult to support healthy aging while consistently undermining one of the body's most essential biological requirements. Sleep optimization does not require perfection. It requires respect. Respect for the fact that humans evolved with rhythms: light and darkness, activity and recovery, wakefulness and rest. Ignoring those rhythms carries consequences. Working with them creates advantages. Nutrition represents another cornerstone of healthy aging. Yet nutrition is also one of the most confusing subjects in modern life. Dietary trends appear constantly. Experts disagree. New studies generate conflicting headlines. One week a food is celebrated. The next week it is criticized. The complexity can feel overwhelming. Fortunately, the broad principles are often simpler than the details. Most longevity research points toward patterns rather than miracle foods. Diets emphasizing whole foods, vegetables, fruits, legumes, healthy fats, adequate protein, minimal ultra-processed foods. These patterns appear repeatedly across populations known for exceptional health and longevity. The exact dietary approach may vary. The underlying themes remain remarkably consistent. Eat foods that resemble their natural origins. Limit foods engineered primarily for convenience and overconsumption. Support the body's biological systems rather than constantly challenging them. No single meal determines your future. But dietary patterns repeated over decades matter enormously. And within discussions about nutrition, intermittent fasting continues attracting attention. As we explored earlier, fasting may influence autophagy and other cellular maintenance pathways. Research remains ongoing. Not every claim is supported by evidence. Not every fasting protocol works for every individual. Yet periods without constant food intake appear capable of activating ancient biological responses that evolved during times of scarcity. The key lesson is not necessarily that everyone should fast. The key lesson is that the body may benefit from occasional opportunities to focus on maintenance rather than constant growth. Humans evolved under cycles: periods of feeding, periods of fasting, periods of activity, periods of rest. Modern life often disrupts those cycles. Restoring them may offer benefits. But healthy aging is not merely physical. One of the most surprising discoveries in longevity research involves relationships, social connection, friendship, community, love. These factors are often overlooked because they do not fit neatly into medical models, yet their influence appears profound. Humans evolved as social beings. For most of history, survival depended on cooperation. Isolation often meant danger. The brain and body still carry traces of that evolutionary history. People with strong social connections frequently experience better health outcomes, lower stress, greater resilience, improved well-being. The mechanisms are complex: behavioral, psychological, biological, all interacting together. What matters is the pattern. Connection appears protective. Loneliness appears costly. The implication is powerful. A meaningful conversation may be doing more for your long-term health than you realize. Then there is purpose, a concept that can sound philosophical until science begins measuring it. Researchers studying long-lived populations often encounter an intriguing pattern. Many people who age exceptionally well possess a strong sense of meaning, a reason to wake up, a role within their community, a sense that their existence matters. Purpose influences behavior. People with purpose often remain active longer, maintain social relationships, manage challenges more effectively, recover from setbacks more successfully. Purpose does not eliminate suffering. It provides direction through suffering, and direction matters, especially over decades. Stress management plays a similar role. Stress itself is unavoidable. Life guarantees challenges, loss, uncertainty, disappointment, change. The goal is not eliminating stress. The goal is learning how to respond to it. Meditation, time in nature, physical activity, meaningful relationships, breathing practices, creative activities. Different people benefit from different approaches. What matters is recognizing that chronic stress is not merely emotional, it becomes biological. Managing stress is not self-indulgence. It is maintenance. Maintenance for the nervous system, the immune system, the cardiovascular system, the aging process itself. Many of these principles become visible in places known as blue zones, regions of the world where unusually high numbers of people reach advanced ages while maintaining relatively good health. Researchers studying these communities expected to find a secret, a special food, a unique supplement, a hidden genetic advantage. Instead, they found patterns: natural movement, strong social bonds, purpose, moderate eating habits, community engagement, lower levels of chronic stress. No magic formula, no miracle intervention, just thousands of behaviors repeated consistently across decades. And perhaps that is the most important lesson of all. Longevity is rarely built through one miracle intervention. It emerges from thousands of daily choices, tiny decisions repeated again and again. A walk instead of remaining seated, a healthy meal instead of a heavily processed one, an earlier bedtime, a conversation with a friend, a moment of gratitude, a workout, a few minutes of reflection. Individually, these choices seem insignificant. Collectively, they shape biological trajectories. The future is not determined by one day. It is influenced by patterns, and patterns become lives. This may sound less exciting than futuristic gene therapies, less dramatic than cellular reprogramming, less revolutionary than artificial intelligence. Yet, these habits possess one extraordinary advantage. They are available now, Not someday, not after the next scientific breakthrough, today. Which brings us to the final chapter of our journey, a question that transcends biology, a question that philosophers, scientists, theologians, and dreamers have debated for centuries. Because after everything we have learned about aging, after all the discoveries, all the technologies, all the possibilities, one mystery remains, and that leads us to the deepest question of all. Even if we could stop aging, should we? Part 12. If humans become immortal, what happens next? Imagine waking up one morning and learning that aging has been cured, not slowed, not delayed-induced, eliminated. Your body will no longer grow biologically older. Your cells will continue repairing themselves indefinitely. Your organs will remain youthful. Disease associated with aging becomes rare. Death from old age disappears. For the first time in human history, the clock stops. What happens next? Would humanity celebrate the greatest victory ever achieved? Or would we discover that immortality creates problems we have never imagined? For thousands of years, human beings have dreamed about escaping aging. The desire appears in myths, religions, legends, literature. Ancient civilizations spoke of magical springs that restored youth. Kings searched for secret elixirs. Alchemists devoted their lives to finding eternal life. Even today, billions of dollars are invested in longevity research. The dream remains alive. Yet something interesting happens whenever humans get close to imagining immortality. Excitement is often followed by uncertainty. Because the question is not simply whether we can live forever. The question is what forever would actually mean. To understand that question, we must first explore the philosophical meaning of aging itself. Throughout this series, we have examined aging through the lens of biology, telomeres, DNA damage, mitochondria, epigenetics, cellular repair, scientific mechanisms. But aging is not merely a biological process. It is also one of the deepest experiences of being human. Every culture on Earth understands aging. Every civilization has built stories around it. A child becomes an adult becomes elderly. Generations rise and fall. Parents become grandparents become memories. The rhythm repeats, again and again, for thousands of years. Aging gives shape to human life. Without it, many of the concepts we take for granted begin changing. Childhood, maturity, retirement, legacy, inheritance, generational wisdom, even the idea of growing up, all are connected to the reality that time is limited. And perhaps this is why philosophers have long argued that death gives meaning to life. At first, that sounds strange. Most people fear death. Most people would gladly avoid suffering, illness, and decline. That is understandable. Yet many thinkers have pointed out a paradox. The fact that life ends may be precisely what makes it precious. Imagine reading a book with an infinite number of pages. No ending, no final chapter, no conclusion. Would every page feel meaningful? Or would urgency disappear? Now imagine the opposite, a book with a limited number of pages. Every chapter matters. Every moment becomes more valuable because it cannot be repeated forever. Human life may work similarly. We cherish time because time is scarce. We treasure relationships because they are temporary. We pursue dreams because we know opportunities are limited. The awareness of mortality shapes countless human decisions. It motivates action. It creates urgency. It encourages courage. It reminds us that every day matters. Without endings, would beginnings feel the same? No one knows. But the question has fascinated humanity for centuries. This leads directly into another powerful idea, the human search for permanence. Deep down, most people want something to last. We want our memories to survive, our achievements, our families, our stories, our influence. Even when we know our lives are finite, we search for ways to leave traces behind. Some people create art, others build businesses, others raise children, others devote themselves to science, teaching, faith, or service. These efforts often reflect the same desire: to contribute something that outlasts us, to leave a mark on the future. Perhaps immortality appeals to us because it appears to solve that problem completely. If we never die, nothing must be left behind. We remain here ourselves. But would that truly satisfy the human spirit? Or would new challenges emerge? Imagine living for 300 years. Would you still feel motivated to act today? Or would every goal become something that could always wait until tomorrow? What about 1,000 years? 10,000 years? At what point does abundance diminish value? These are not scientific questions. They are philosophical ones. and they reveal how complicated immortality may be. Many people imagine eternal life as a gift. Others imagine it as a curse. Both perspectives contain interesting arguments. Supporters of immortality often point to obvious benefits: more time with loved ones, more opportunities to learn, more experiences, more knowledge, more creativity. Imagine becoming a master of multiple sciences, learning dozens of languages, witnessing centuries of human history, watching civilization evolve. The possibilities seem limitless. A longer, healthy life could unlock extraordinary human potential. Great discoveries might emerge from minds given more time. Relationships could deepen, wisdom could accumulate, entire new forms of culture might develop. From this perspective, defeating aging appears overwhelmingly positive, yet critics raise equally fascinating concerns. What happens if people stop making room for future generations? How would societies evolve? Would innovation slow if the same individuals remained in positions of influence for centuries? Would population growth become unsustainable? Would inequality increase if longevity technologies remained available only to the wealthy? What happens to ambition when there is no deadline? What happens to meaning when there is no ending? Again, nobody knows, because immortality remains hypothetical. What science has actually achieved is far more modest, and understanding that distinction is important. Despite exciting headlines, science has not cured aging. No laboratory has discovered immortality. No treatment can stop biological aging entirely. No human being has demonstrated indefinite lifespan. The oldest verified human lifespan remains just over 120 years. What science has proven is something different. Aging is not a single process. It is a collection of interconnected biological mechanisms. Many of those mechanisms can be influenced. Some can be slowed. Some may eventually become partially reversible. Researchers have successfully extended lifespan in laboratory organisms. They have identified important cellular pathways. They have developed promising therapies. They have uncovered remarkable insights into how aging works. These achievements are extraordinary, but they do not yet amount to immortality, not even close. And this brings us to what science still does not know. Can human lifespan be dramatically extended? Possibly. Can aging eventually become a treatable condition? Perhaps. Can biological age be reversed safely? Maybe. Can humans live to 150 years? Some researchers believe it is possible. Others remain skeptical. Can immortality ever become reality? At present, nobody knows. The truth is that aging remains one of the greatest mysteries in science. We understand more than ever before, yet enormous questions remain unanswered, and perhaps that uncertainty is what makes this field so fascinating. We stand at a unique moment in history. For the first time, humanity possesses tools capable of investigating aging at the molecular level. Artificial intelligence is accelerating research. Genetics continues advancing. Cellular reprogramming is opening new possibilities. Longevity science is attracting unprecedented attention. The future may look very different from the past. Children born today may experience lifespans that seem remarkable by current standards. Medical breakthroughs may transform how aging is understood and treated. The coming century could reshape humanity's relationship with time itself. But even if those breakthroughs arrive, one question will remain. What is the ultimate goal? Is the goal to live forever, or is the goal to live well? Perhaps that distinction matters more than we realize, because when most people imagine a longer life, they are not dreaming about endless years of frailty. They are imagining vitality, health, energy, curiosity, connection, meaning. They want more life in their years, not merely more years in their life. And perhaps that is where science and philosophy finally meet. The purpose of longevity research may not be escaping death entirely. The purpose may be reducing suffering, extending health, preserving independence, allowing people to remain fully engaged with life for longer. Perhaps the goal is not to live forever. Perhaps the goal is to remain healthy, curious, and fully alive for as long as possible. Because when we step back and look at the bigger picture, aging itself begins to appear differently. A wrinkle is not merely a sign of decline, it is evidence of years lived, challenges survived, experiences accumulated. A gray hair is not simply a marker of time passing. It is proof that time was given. Every wrinkle tells a story. Every gray hair marks a victory against time. The real miracle may not be escaping aging. The real miracle is that life exists at all, that atoms somehow became cells, that cells became organisms, that organisms became conscious beings capable of wondering why they exist, capable of studying the universe, capable of asking questions about aging, mortality, and meaning. Perhaps that is the greatest mystery of all. Not why we age, but why life emerged in the first place. And so we arrive at the end of this journey. We began with a simple question: why do we age? Along the way, we explored telomeres, zombie cells, mitochondria, DNA damage, epigenetics, autophagy, longevity research, the future of medicine, and ultimately the meaning of mortality itself. What we discovered is that aging is neither simple nor inevitable in the way people once believed. It is a complex biological process, one that science is beginning to understand with extraordinary precision. Yet understanding aging does not automatically solve the deeper mystery of how to live. That answer remains personal, individual, uniquely human. In every second, trillions of cells are fighting to keep you alive. The question is not whether time is passing. The question is how you choose to spend the time you are given. And if you enjoyed this journey through science, aging, and the future of humanity, please consider liking this video and subscribing to the channel. Your support genuinely helps this growing channel reach more curious minds who love exploring the biggest questions in science and philosophy. And I would love to hear your thoughts in the comments. If science could stop aging tomorrow, would you choose immortality? Why or why not? And if you know someone who loves science, philosophy, longevity research, or the future of humanity, share this episode with them. Thank you for spending this time with me. Until our next exploration, stay curious. And remember, every moment you are alive is already one of the rarest events in the universe.